Hello guys, so good morning. I am Andres and I am the last reporter. So for my part, I'm going to tackle about how do nets protect the host and how can they contribute to tissue damage or thrombosis. So first, let us define what are nets. So nets stands for neutrophil extracellular traps, and these nets composed of a network of decondensed chromatin coated with nucleoproteins like the histones and specific granulosa proteins such as the neutrophil elastase and myeloperoxidase. Nets are termed to be double-edged sword because it serves two functions, which is for protective and harmful. Sa protective nga part, ang purpose sa nets is for host defense because nets captures and eliminates pathogens, preventing their spread throughout the body. And for the harmful part, which is the promoting thrombosis. To define, thrombosis is a dangerous formation of a blood clot or thrombus inside a vein or artery, obstructing normal blood flow. In this figure, this is an illustration of what the net looks like. So in the first photo, it is a neutrophil net, which is the color green that caught a bacterium. Now the second photo, it is the process of the net formation, and later we're going to further discuss that. And on the last photo, which is the bacteria, which is color red, ensnared on a neutrophil net. Nets are formed through the process called netosis, which is a programmed neutrophil death which they kill their own self, a form called beneficial suicide. This process is important for nets to be able to release web-like structures to capture more pathogens that are not engulfed by neutrophil itself to ensure nga wala gyoy mabilin. And once again, the release webs are composed of histone, specific enzymes such as neutrophil elastase and myeloperoxidase. So for short, kibali like sa lodo gid ta aning nets kay nagpakamatay gid sila just to save and protect us, and the main cause of its activation is through infections, inflammations, or damage signals. This process is necessary good for immune defense, pero pag sobra-sobra na pud ang production, it can contribute to diseases like thrombosis. So now let us going to dive deeper behind the mechanism of nets formation. There are two kinds of formed nets, which is divided into suicidal netosis and vital netosis. Magmatter asa sa duhaan ang ma-form depende sa nature of stimulation. Karon, i-focus nato is ang suicidal netosis. So, ang one of the examples of specific molecules and substances that stimulate suicidal netosis are 4-bol-12 myristate, 13-acetate or the PMA, pro-inflammatory cytokines, chemokines, and antibodies. Karon, the process of suicidal netosis begins at the cell surface or through direct stimulation. For this example, PMA ang nag-trigger. Ang PMA, which is ang atong stimulus karon that activates PKC or the protein kinase C. And kana si PKC, i-turn on na niya si NADPH oxidase complex or the for short NOx that produces ROS or the reactive oxygen species. Then next, ang ROS i-trigger dayon niya ang RAF-MEK na signaling pathway, og ang purpose ana niya serves siya as coordinator telling the rest of the cell to begin the dismantling process. And next, ang migration of the two key enzymes, the elastase and myeloperoxidase, leave their storage granules and enter the nucleus. And at the same time po, PAD4 or the peptidylarginine deaminase 4 is activated. So, ang purpose ana niya ni PAD4 is i-modify niya ang histones nga nag-wrap around sa DNA to spools the DNA through a process called citrullination. So, ang result sa citrullination makes the DNA lose its tight grip and start to expand or decondense. Next is the nuclear rupture na dayon, which as the DNA expands, the nuclear envelope breaks down. Now, so once ni rupture na ang nucleus, the decondensed DNA ug ang may antimicrobial proteins, ang elastase ug myeloperoxidase magmix na sila together inside the main body of the cell or the cytoplasm. And ang ending dayon ana nga process is the extracellular release, where the expansion of the DNA makakreate siyag immense internal pressure that results for another rupture or lysis. So lysis naman, namatay na ang cell or ni-explode na gid ang cell, releasing the nets na outward. That results a sticky toxic web of DNA and proteins that can trap bacteria na dayon. Diri na pud ta sa vital nitosis. Naagi significant difference ang duha ka types of net. Unlike suicidal nitosis, vital nitosis can occur without NADPH oxidase activity, meaning it doesn't rely on production of reactive oxygen species and capable of survival pud siya because the outer membrane remains intact. Then the neutrophil can continue its traditional defense roles na dayon, such as scrolling, chemotaxis, and phagocytosis, which stays alive lang gyud siya, unlike sa suicidal nitosis gyud nga malysis. Same gyapon sa suicidal nitosis, first is recognition sa stimulus. Then our example diri is LPS binding to TLR4. Pero pwede ra gyud gihapon ini asa sa duha ang mobind sa TLR. Pero atong example diri is LPS or lipopolysaccharide. Then as what you can see, pwede gihapon nga ang complement protein mo bind sa neutrophil complement receptor. Kay kung kagaina ang stimulus sa suicidal nitosis is katong PMA, cytokines, chemokines, and antibody. Ang sakaron sa vital nitosis is activated platelets, microorganisms, ug ang complement proteins. To proceed, lipopolysaccharide binds to the TLR4 receptor on the neutrophil's surface. The binding sends an immediate signal that a biological invasion is occurring. Then it will result to TLR4 activation. Ang purpose ng TLR4 activation is to bypass the death program and trigger a survival-based defense. Unlike chemical triggers like PMA that lead to cell death, TLR4 activation initiates a signaling cascade that works without needing ROS. So in turn, this keeps the cell's internal structures safe while preparing the DNA for release. Next dayon is ang PAD4 activation. Just like in the suicidal version, PAD4 citrullilates histones. This causes the chromatin to lose its tight structure and decondense. PAD4 activation prepare the DNA to be moved out of the nucleus. This decondensation is more controlled. Next is the calcium influx. It acts as a secondary messenger that activates specialized channels to turn in the SK3 potassium channels. SK3 activation serves as a regulator. This allows the neutrophil to package its DNA into small molecules that sprout from the nuclear membrane without causing the whole cell to swell and burst. Next is the net release na dayon to export the traps while keeping the neutrophil's function running. But the traps are stored in vesicles that move to the outer plasma membrane and are secreted into the extracellular space. This results the neutrophil to lose its nucleus but remains alive and active. It can still move and phagocytose bacteria, providing a continuous defense alongside the traps it just laid. Net cytotoxic nature causes tissue damage, which the histones can cause blood vessels leak and can lead to cell death, and to enzymes that kills pathogens but break down surrounding structural proteins if overproduced. And third, an excessive production sa nets fuel cytokine storm. Next slide. The progression of netosis lead to thrombosis as nets catches RBCs and platelets, enlarging the clot. Nets also activates factor 12, which furthermore starts the clotting cascade. But at the same time, nets block anti-clot system that keeps clot growing without stopping, as well as nets are capable of activating platelets that enable platelets to signal neutrophil to make more nets, thus therefore results in clot keeps getting bigger and stronger, a term called vicious cycle. And lastly, number 5, normally body uses plasmin or drugs like TPA to break clots, but nets form a DNA barrier protecting a clot from being destroyed, thus leads clots become resistant to treatment. And on the last slide, nets are clinically important because they are involved in several serious diseases in deep vein thrombosis and pulmonary embolism. Nets promote excessive blood clot formation by trapping platelets and activating clotting factors. In sepsis, nets are released in large amounts during infection, causing tissue damage. In COVID-19, nets contribute to cytokine storm, where the immune system becomes overactive, leading to severe inflammation, clotting problems, and organ damage. Overall, nets can worsen disease by increasing both clot formation and inflammation. High levels of extracellular DNA, MPO, and citrullinated histone in the blood serves as biomarkers of nets, indicating increased risk of thrombosis and can also suggest more severe disease or higher mortality. And lastly, current treatments are being studied to target nets, which DNase 1 helps break down nets, while PAD4 inhibitors prevent their formation. These approaches may help reduce or prevent blood clots. So that would be all. Thank you.